This right here is a rod. And a rod is kind of a weird cell because typically when there's some kind of stimulus, that's going to cause a cell to do a thing, right? So, but with rods, they actually work in the absence of light. So you can see here it says dark, and then down here it says light. What we're going to see is that when there is no light, that's when rods are active. And so when light occurs, when that stimulus, when a photon actually hits it, that's when the rod is going to turn off. It's a really, really fascinating process. Now, to understand and learn this, we're going to be using one of Kenhub's brand new study units on physiology. Now, this one, scanned to the top, you can see is all about vision. Now, this is a premium feature, but we load these with so much value, it is absolutely insane. You can see we have videos, we have all these images. And by the way, when you sign up for Kenhub, you can get access for free to all of our images, all of our articles. They're in the thousands. But there is so much that we pack into these lessons, these study units. We're going to go ahead and leave a link down in the description below to a separate study unit on reflex arcs that I strongly encourage you to go check on out. So again, go find that in the link in the description below. But we're going to be figuring out how this works. So I'm going to hit this magnifying glass here. And before we actually look at this image, I think it's probably best to discuss a little bit of the microanatomy of rods and a little bit of cones too, because they're very similar. Right, so when we talk about rods and cones, these are the photoreceptor cells. Right? So what that means is they detect the presence of light. And what they're going to do is then be able to translate that, obviously all the way down the line when we get to the visual cortices in the occipital lobe into vision. But rods are what we call scotopic cells. So basically what they do is it's low light sensitivity. It's probably the best way to put it. Right? So they're going to be able to detect and help you see in low light situations. Where cones, on the other hand, they're going to be all about light. Cones are also where you're going to see color. So rods, not so much color. Cones, that's where you're going to see color. Although it's not that. That's a little bit generalized because again, it's more low light than light, but still, I think that works. Now, the main things we want to talk about here, we're not going to go over all of these here. It's just, there's a couple things we want to understand. So first off, you can see that in here, we call this the outer segment. And the outer segment is where pretty much everything we're going to be talking about is going to be happening. And then you have to understand that it has all this other stuff like mitochondria. The mitochondria are going to be essential because it requires a lot of energy to actually turn, like this cascade that we're going to be talking about. It's going to just require a lot of energy to be able to, down here at the synapse with the bipolar neuron, to just continue that signal and convert everything we need to. But you basically have this outer segment. You have this inner segment with the mitochondria. You have a cell body. And then at the end, you're going to see what we call, it's a little bit different depending on, right? So like a rod cell, we call this the spirule. And then with a cone, we call this the pedicle. Right? But what's going to happen is in here, there are the synaptic vesicles, and they're going to be releasing neurotransmitters to another neuron on either side, which is called a bipolar neuron. So real quick, outer segment, this is where most of what we're about to talk about is going to be happening. And the goal is then to, once you detect the light, that is going to cause something to happen down here at the synapse with a bipolar neuron, right? So again, but the other thing to understand is inside of here, you see all these little things? We call these membrane discs. And these membrane discs, there are going to be hundreds of them inside of these outer segments. Now, we're going to be focusing on rods today, but a very similar thing happens inside of cones. But I think it's just going to be easiest if we focus primarily on rods. So what I want to do is now go back to this image that we started with. And this is really, really cool because what you're seeing here, now we can kind of see how it's labeled. Where it says outer segment and then extracellular fluid, what you're looking at here, this like gray that you're seeing, well, that's the outside here. So, and then the extracellular fluid would be right here. So basically we're in the retina. The retina is the photoreceptive layer in the eye. So it's the most internal aspect of the eye, and that's where you're going to have all the sensitive cells. So just picture outside of the rod is going to be a bunch of fluid, extracellular fluid, right? So then here's that outer segment, just like this, and then here's the membrane disc. So we go back here, you can see where it says disc. That's what this is showing here. So you're looking at one of those lines right here. So imagine we're zooming in. Let's just say we're zooming in right here. And that's where you're going to see this membrane disc. And now we see the outer segment and the extracellular fluid. Now, first thing I want you to look at is this right here. All right, so this is an ion channel. And what's happening is if you look really closely, you're going to see that sodium ions are flowing in. Right? So there's a bunch of sodium that's on the outside of the cell in the extracellular fluid, but this ion channel is open, and that means the sodium ions are going to flow inside of the cell. Now, these are positively charged sodium ions. And so what that means is that that is going to depolarize the cell, which basically just means it's going to be more positive internally, and that is going to cause an action potential to fire. So what I'm saying here is, there's, in the presence of dark, or rather in the absence of light, sodium is constantly flowing into the cell. And when sodium is constantly flowing into the cell, that is going to change the voltage internally. So it's around negative 40 millivolts. Now, a resting potential for a neuron is negative 70 millivolts. So by increasing the voltage here, all of a sudden that causes a depolarization and you send an action potential. So I want you to picture this, right? Sodium is flowing inside of here. That changes the net charge inside of this rod, and that's eventually going to lead to an action potential being released. And then down here in that rod spherule, that is where we're looking at this, right? This is, and you can see here it says GLU. This is glutamate. Glutamate is a neurotransmitter. Right, so what's happening, as long as the sodium is flowing in, glutamate is going to be released at the synapse between the rod and the bipolar neuron that's going to be in here. Right? So this is what is just happening in the darkness, right? So you close your eyes, or it's nighttime or something like that. Just sodium is just flowing into the rods, and this is going to tell your brain, hey, there's no light here, right? There's an absence of light. So then the question then becomes, well, what happens when light hits it? So that's where we have to look here. So what you're seeing here, it says rhodopsin. Now, in a cone, you could call this conopsin. That's one name, or iodopsin. I think it's iodopsin. Yeah, we can see even here, iodopsin. I've always called it conopsin. So, but they're very similar molecules. So this is actually two things, right? So rhodopsin is made of what is called opsin, which you see in green. And then inside of it, you can see what's called retinol. And so specifically this shape of it, you see how it's bent here? This is what we call cis retinol. So the cis part means that it's bent like this. So rhodopsin is actually the combination of opsin, which is the green, and retinol, which is this shape that you're seeing here, this little bent with the tail, right? So next to them is something called transducin. Now, transducin is actually all inside of here. There's a bunch of transducin. Now, transducin is made of various different subunits. So if you look really closely, you're going to see it'll say alpha, gamma, and then beta subunits. And so they're all kind of attached. But one thing that's really important to understand is this transducin, you have to look really closely, it is not touching the rhodopsin. This is what is happening when there is no light. Opsin and retinol are kind of like hanging out. Transducin is hanging out. Nothing incredible is happening. So then what's going to occur is we're going to go down to this image. And we can see it says light. So imagine a photon, right? A particle of light is going to come in here. And what it's going to do is it's actually going to hit that retinol. And when it hits the retinol, it's going to cause a conformational change, right? It's going to change shape. And instead of being cis retinol, it is now going to be trans retinol. And so now you can see that its tail here is straight. And this is everything, because once this occurs, this is what we're talking about when we say like phototransduction cascade, right? So in the presence of light, a whole cascade of things is going to happen. The only thing that was happening in the dark is sodium flowing in, but once light hits, boom. What's going to happen is opsin is going to change its shape. Not only is cis retinol going to turn into trans retinol, But opsin will change its shape and it will bind. If you look really closely, we can see that transducin is now attached, right? It has bound itself to opsin. And when this occurs, well, actually, let's say this, because you're probably not going to be able to see this because it's really, really tiny, but there's something on here called GDP. So GDP is actually present here as well. GDP is just hanging out on this thing that the one we want to look at is one that looks kind of like a bean. This is the alpha subunit. GDP is attached. You can see that here, this little red thing and these two little things. When GDP is attached to the alpha subunit, nothing is happening, right? So that would be happening here. But when light hits the rhodopsin and opsin changes its shape and transducin attaches to the opsin, what will happen is the alpha subunit here will let go of GDP. It just drops it. And when it drops it, there's another molecule in the area called, and again, you're probably not going to be able to see this, called GTP. And I want to be really clear here because I've seen this mistake a lot. A lot of times people think that when light hits rhodopsin, it causes some kind of conversion from GDP to GTP. That's not the case here. GTP is all over inside of the intracellular fluid. All right, so there's a bunch of GTP there. But what happens is there's nowhere for the GTP to go because the spot it wants to take on this alpha subunit is occupied by GDP. But remember, when light hits the rhodopsin, an opsin changes its shape and transducin binds to the opsin, it causes the GDP to be released. And what'll happen is GTP attaches, it binds to it. And when that occurs, the alpha subunit breaks off from the gamma and beta subunits. And it drifts down to what's called phosphodiesterase. And that is going to cause, so this is an enzyme. Now this enzyme, we can see it up here. So it says PDE. Phosphodiesterase is going to be located in that membrane disc. So it's bound to the membrane here. Do you see that? So once this becomes activated, this enzyme, it is going to come over to this thing called cyclic GMP, right? Cyclic GMP, we can see that it is attached to this ion channel. When cyclic GMP or cGMP is attached to the ion channel, the door is open. And all of these sodium ions are just going to flow into the cell. And then again, we get a depolarized cell. But when that alpha subunit with GTP comes down and activates the PDE, the phosphodiesterase, that is going to convert cGMP to GMP. And when you do that, that reduces the overall intracellular levels of cGMP. And by doing that, you close the door. Now you don't have this ion channel open, and we can see the sodium ions are stuck inside of the extracellular fluid. So do you see what's happening? This is the cascade. This is that phototransduction cascade. Light causes transducin to release the alpha subunit, which will activate PDE, which will then convert cGMP to GMP, which will close the door. Sodium ions can no longer get in, and what we end up with is a hyperpolarization of the cell. So if you remember, sodium depolarizes it. It goes down to, or up to, negative 40 millivolts. But when you close the door and sodium can't go in there, all of a sudden, the internal environment of that cell becomes more negative. And what that means is less glutamate is going to be released at that synapse. It's a cascade of events. And what that then tells the brain is, oh, there's light. All right, it's a complex process, yes, but it's really, really cool when you think about it, right? In the dark, sodium comes in. In the light, sodium can't come in. And this is the whole process that is happening here with rhodopsin, and a very similar thing is happening with conopsin inside of a cone. And all of this is going to result in you not realizing that there is or isn't light. Absolutely fascinating. And remember, this is just one part of our vision study unit. And we're going to go ahead and leave a link down in the description below to a reflex arc study unit. That's one's all about like reflexes. You hit the knee and it kind of goes like this. There's other reflexes like stretch reflexes and Golgi tendon reflexes. There's all sorts of really cool stuff that we were going to want you to see. So be sure to check that out. Now, if you enjoyed this video, be sure to hit the like button, subscribe, comment. All those little tiny things actually go a long way in helping these videos perform better. But I just want to say thank you for hanging out with me, and I will see you in the next one.